Is that okay?
That's okay.
Okay. Just let me know if you are ready.
Is everyone ready?
Yeah, okay,
No,
sure.
okay.
Dr. Mochi.
I'm so sorry. I will not be, I cannot be there.
Yeah, it's okay.
But
No problem.
suddenly, early morning, I got something in my
Yeah,
gastrointestinal tract.
yeah, it's okay. Thank you for the online course
Yeah, sure, of
today.
course, yes,
Yeah,
yes. It's my responsibility, so I'm very happy to do that.
yeah.
Well, of course, you are interested in analyzing the microbiome. Then last time I mentioned that you have to get 100% of microorganism in your from your sample. In my case, my sample is intestine, maybe your case, food, but it doesn't matter. So you should get entire microorganism in order to understand, or in order to investigate microbiota. Then you also have, I also told you that to get or extract DNA from gram-positive or gram-negative bacteria, sometimes it's very difficult. You remember that, particularly from gram-positive bacteria. So what kind of kit uh, animal studies or laboratory studies, not computer science only studies, bioinformatics studies. So we are collecting sample once again. Then what kind of system we use? This is also another option. Then also what kind of reagent will be used for storage of DNA sample? You have to consider, think about it. Then which region of 16 years ribosomal RNA will be targeted? It's another option. Then we are usually sequencing V3 and V4. This slide just showed V4, but in our analysis, V3 and V4 are regions usually sequence, then perform sequencing analysis. We are basically using Illumina, but you are using different company-based sequencer, but it doesn't matter. I'm sure that it's fine. Then once you get sequence data, then what's happened? What you do? So this is second training course. Then another thing that I told you is that the NGS is powerful tool to sequence lots of DNA sample on the same time. So usually in our studies, we are sequencing 50,000 or 100,000 DNA from each sample. Then we combine everything. Then we sequence all together. That's why in your case, we mix right from 200 to 500 sample on the same time in the same run. Then cost is like this in Japan. So you can just calculate. So this is the thing that I told you last time. Then maybe you are more interested in analyzing the data. So I thinking that it's better for you to show you about exact data that we have, then maybe show you what kind of study we did, then how we collect, then how we analyze. So this is the step. Then if we don't have any data in the presentation, then just principle of the training analysis course. So maybe it's not interested in for you. So this today's data is very unpublished, and I'm still writing for submissions of very new data, but I'm very happy to introduce. But this is the concept. Maybe some people had a chance to join in my general lectures in the first day. then I am analyzing the relationship between microbiome and also mammary gland immune functions. Then what I told you in the general lecture is that like immune cells, like producing antibodies. This is my research target. So antibodies that really baby really needs to get it from the milk. But how antibodies is produced in mothers during lactations, then I mentioned that the cells producing antibodies are coming from intestine, which is very long distance. But mother really wants to use intestinal immune system to produce antibodies. That's why mothers needs to send the cells producing antibodies from intestine to the mammary gland during lactation. This is one finding. Then also we discovered that during lactation, some particular microorganisms stimulate gastrointestinal immune system named Bacteroides acidophacins and Prevotella buccaris, both of which have potential functions to stimulate immune cells in the gastrointestinal tract. So if mother have this, then immune cells are stimulated by these bacteria. Then immune cells move from intestine to the mammary gland. Then cells produce antibodies. So this is a concept. Then we are now doing the next stage of the experiment. So this is the sample in NGS studies that I really want to introduce. So this is most important. Actually, this is not like database or bioinformatics studies, right? But if you don't understand this research concept, then you will not understand anything. So please try to understand what we are doing in vivo. Then please try to compare your experiment. I'm sure that the research is totally different, but you have exact design of the research, then you want to compare something like before, after fermentations. So comparison, we always have a research aim to compare something and something. So in our experiment, what we are interested in comparing, so this slide show you. So we are very interested in understanding how milk-derived antibodies works in the baby. Once again, how milk-derived, we say maternal milk, mother-generated milk works in the baby via breastfeeding. Then people believe that milk is very important because if baby do not have milk antibodies, then baby easily get infections. But this is true, but it's very like not clear concept. So how antibody works? Well, what kind of functions actually milk antibody has in baby, in offspring? So this is research concept that we are interested in. So in this case, we normal mothers. This is normal experiment. But also, maybe you have never heard about this skit, but I want to explain. We prepared skit mice. That is immunodeficient mice. They do not have any immune cells. So meaning that we maintain these mice in the very clean facilities, then they can stay grow up very well without any in the animals. So once again, we prepare normal mice, and also we prepare skid mice to understand the function of milk immune system or milk immune factors or milk antibodies. So most experiments, we use next generation like kids, babies, Y-type baby and skid babies. But most important design in this study is that actually these mothers, Y-type mothers and skid mothers, they get baby on the same day. They get baby on the same day. But half of the baby from wild-type normal mice and from immunodeficient skid mothers switched just after birth. Like, then notice that my baby changed. But mothers or mice do not recognize even if we change the baby. So this is, we say, cross fostering models. Half of baby change to the others, then half of change baby to change to the others. Then why we change is because these skid babies, they do not have immune cells because this is skid baby. Baby also do not have immune cells, but they can drink wild type normal mother milk. So even if they don't have, baby do not have immune cells, but they can get antibodies from the mothers. The other case is this. So they are very normal mice, then their immune system are completely normal. But during lactation, their milk is from skid mice. So meaning that they are not they cannot take milk antibodies from the mothers. Then we will compare what's happened. If we have milk antibodies or if we do not have milk antibodies, what's happened? Particularly microbiome in the baby. Then our experiment designed 2 times, like before weaning, like during lactation, exactly drinking milk. And after like 10 weeks after birth, they already develop. Meaning that we really want to understand the effect of the milk antibodies even after weaning or before weaning. So I actually want to stop this because if you don't understand this experimental design, then you and I will have problems. So if you have any question, let me know.
Excuse me. I think the slide is still the same, or we only can see the same slide.
Oh, it doesn't move?
It doesn't move.
Oh, it's problem, it's problem. Which side? First page?
You can click the share icon. Can you click?
Okay. Stop. Oh, I'm so sorry, I didn't know.
It's okay. We
So,
can hear you.
okay, I will do one more time.
Ah,
Can you see?
yes.
It works? Next. Oh, it doesn't move. Next page. Okay. Okay. Then both mothers get baby on the same day. So baby also have normal immune system in the normal mice. So what WT means wild SCID is immunodeficient. So SCID baby also do not have immune cells. But on the same day, once they get baby, then half of the baby, half of the baby changed like this. Then this is gray animal is skit baby, but they drink normal normal mice derived, even though they do not have their immune cell, but they can get antibodies from milk during. Then our research interest is also to investigate how much difference, like before weaning, but actually during lactation, or even after weaning, how much impact of the milk antibodies in milk for next generation, next offspring. So we still keep for additional 7 weeks at 10 weeks of age. Then we analyze 4 mice, 4 groups, and also we analyze 4 babies, 4 group babies at 2 different time points. Is that okay? Okay, so try to compare your experiment. It's okay. It's experimental design totally different. I agree. I know that. But we have some kind of comparison. Then we use 60S ribosomal RNA. Then how we analyze, then how we get the data. So this is a training, today's training course. But before that, please predict, hypothesize that what's happened. If it's very interesting for you, hopefully, because if we don't have like antibodies from the mom, then particularly after maturation, after development, the problem is still continues forever or not, but just during lactation or even after weaning. That's the question. Then if like the concept, hypothesis, like even if after like weaning, like this time point, then if the baby, normal baby, have problem, if they receive milk which do not contain antibodies, then we have to think about how we give milk. Like people, in usual, most people use artificial milk. So how we think about the immune cell, like or antibodies, should be included in the artificial milk for the babies. So this is a concept. Okay. So to address these questions, then we have several comparisons. So first comparison is normal and skit wild type. This means that the try to see this wild type, small word. So this means baby, mother, baby, mother, baby, mother, baby. Then we are analyzing all baby. Once again, we are analyzing all baby before, after weaning. So their phenotype, genotype is normal wild type and normal wild type. But only problem between these two groups is that this is okay because their milk contains antibodies, but he or she has problem because their milk did not contain antibodies because mother is sick. And this is very straightforward comparison because white normal, skid. So it's like very good animal and very bad animals. Then this is comparison like normal and normal skid. So if babies do not have any like immune system, but if baby get milk which contain antibody, what's happened? So this is a comparison. And this is additional comparison. Normal skid. So this is baby are immunodeficient, but if immunodeficient baby get antibodies from the normal milk, what's happened? Then this is additional comparison, skid normal, skid. So meaning that if like the milk do not, which it doesn't contain milk, but if the baby is mothers cannot produce milk abundantly, then in this case, mothers buy artificial milk. What kind of immunity like antibodies are contained? So this is almost a similar situation. So like if the mother or if artificial milk do not contain milk antibodies, then what's happened in the healthy baby? So this is very important comparison. And this is very powerful comparison. Normal, immunodeficient. We should see something different. And the other comparison, you don't care about. You don't care about it. Okay. So everything okay. So I don't explain more about our experiment because which is not related to your field, but just try to compare your study. So we have many animals, like in this case around 10. So more than 15 animals were used in these studies. Then meaning that we have like more than 50 samples or animals derived data were collected. I forgot to tell you that. But our target is both small intestine and large intestine. Small intestine and large intestine. So we collected small intestinal sample and large intestinal sample from individual group. So if we have 50 animals, then we got 100 sample from small and large intestine. Then we perform single S ribosomal RNA studies. Then this is very low data. But this is not entire data because we have 100 data, okay? But just summarize to tell you how much impact on male using this Excel file. Then this is a genus level of microbiome. Then what we have shown that we have detected these genus. Then how much percent of, for example, this biophilia were detected? And this is still average, but that is very few, like 0.04 or something. Next one is 0.2. But the other one is bacteroides, which is 4.1 or something. But this is, again, sorry, average or standard deviations. Then in some case, we see not a sign. But we have a lot, but you don't surprise. Because this is genus. Then genus is very bottom or low complicated criteria in the microbiome. So if you go like family data, phylum data, then most of phylum has been categorized, can be categorized. So if you go deeper, in some case, bacteria does not have name in genus, the data will be identified as not assigned. So this very usual cases. Then we will focus like the data identified as certain genus bacteria. So here's maybe 25 genus are listed. Then we'll try to understand what's happened. So in the microbiome studies, then people like to use like this bioinformatics tool. Then have you heard this chime? We say chime. I'm sure pronunciation in Indonesia, but this is well-known omics science tool for microbiome studies. Then this is completely free. So you can link Google Chime 2, QIIME2, then just learn how to use it. But this is very standard model platform to understand. So once we get the sequence raw data, then we basically use the data and subject to this Chime2 pipeline to understand. Then Chime2 provide us like alpha diversity and beta diversity. So using this, then you can precisely appreciate what kind of data are obtained briefly. Then this brief mapping is very, very important to think about what kind of comparison you want. So try to get this brief data using Chime2. Just load it, your data, then type in some calculation, then you can get it. So this case, in our case, so we have so many data, then one plot means one sample. Once again, I have two samples, small intestine and large intestine from each individual mouse. So two plot means from same animal. But again, so we have small intestine, SI, small intestinal sample, and large intestinal sample. This is the microbiome profiling. Then maybe I already show you in the general lecture course. If plot is very close, meaning that microbiome is very similar. Then if plot different, completely different, then microbiome is totally different. So how you see the data from this initial beta diversity profiling? So you try to surprise because the difference, because we have small intestine at 2 weeks of age, 10 weeks of age, then we don't divide anything like normal, 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 skin, skin, normal, skin, skin. Just combine everything. Just divide it into different stage, 2 weeks, 10 weeks, and different location like small intestine and large intestine. So 2 weeks is before weaning, 10 weeks after weaning in mice. So try to surprise how much difference of the microbiome in the small intestine before weaning, between before weaning and after weaning. So you can see huge changes of the microbiome in the small intestine at 2 weeks and between 2 weeks and 10 weeks. So meaning that milk have very strong impact on developing 2 weeks type of the microbiome in the small intestine, and 10 weeks, and then after that, the microbiome in the small intestine dramatically changed. Then how about large intestine? So large intestine is this. Actually, clear a bright blue and dark blue. This is the difference between 2 weeks and 10 weeks. So you can see slight difference then maybe you'll understand how much difference in the microbiome of the large intestine before and between 2 weeks and 10 weeks. But maybe you'll also understand if we do not, combine all samples, then if we just analyze fecal sample like this, large intestine or fecal sample, then your difference is only these areas. But if you combine, if you add all data, then you see more large differences. So what kind of sample will be used? It's up to you. Then some studies, people just use very small comparison groups. And then some people use very samples which are totally different. But I would like to emphasize that you at least have to understand what scale of studies you are trying to do. Otherwise, you have possibilities of getting like estimated data. It's very dangerous situations. So in my case, in your case, between small intestine and large intestine, or between before weaning, after weaning, is very good comparison to understand the entire microbiome. Then we really want to understand how much impact of milk derived immune factors in the babies. So this is more deeper analysis. So try to understand. So maybe this kind of data, you have a chance to see some papers. So these data, all data are from China, this. So you can use this platform for creating this kind of general data. And this is alpha diversity and beta diversity and some statistical analysis. So what we did is that we really want to understand how much impact of milk immune factor like antibodies. Then I realized that we should break down the group. We understand entire differences. Then we should break down to understand the each tissue at each time point to precisely understand. Otherwise, because it looks like this one little bit different, but very close compared with this. So we have to break down the data to understand each time point and each like tissues will be target. So maybe you'll also imagine your experiment like what kind of sample, like before fermentation, after fermentation, what kind of part of food will be. It's same thing. So try to get as much as possible. Otherwise, you have a chance to have very overestimated data. It's not good for you. So again, so this is small intestine. Wild type, this is completely normal. Wild type skid, normal mothers and skid, immunodeficient baby. Then we analyze baby. Then wild type baby get received immunodeficient milk, which is not containing antibodies, and this is very much abnormal model skid. Once again, we analyze baby. Then this is phylum level, very large scale, family level, little bit complicated, then genus level, much more complicated. Then from data. So this is average. So this you maybe we create this. I'm sorry, this is not from China. So this is just we created by ourselves. to show you what type of bacteria obtained. But using CHIME, the CHIME do like database matching. So if we have data, sequence data, then CHIME provide us what kind of bacteria we have. So these Excel data are from CHIME. Then using this Excel data, then I want to highlight what kind of bacteria basically we have. But again, this color, this is not probably not every bacteria, because we have lots of bacteria here. Then we still have minor ones. So wild type baby, skid mother derived by skid. Then we analyze baby. So maybe I already told you. I don't remember. I explained the difference. Once again, diversity is how much diversity we have. Like you and I. So try to understand. Then the data from the small intestine at 2 weeks of age during lactations, I do not see any differences in alpha diversity and beta diversities, even statistical analysis. Meaning that this is kind of surprise data, because we try to, this stage, we try to see the impact of maternal milk antibodies during lactations. Then we found that small intestine is very unique. But even if mothers can make antibodies, healthy babies, doesn't matter, don't care about it. So maybe artificial milk, if nutrient is sufficient enough without antibodies, but they are growing in small intestinal microbiome, they don't have any problem at all. So this is the data I show you. 3 more data, like small intestine at 2 weeks, small intestine at 10 weeks, large intestine at 2 weeks, large intestine at 10 weeks. So try to follow me. So this large intestine, then this is alpha diversity. Then something happened, because we will see some differences. Like then I'm super surprised, because wild type mothers, then wild type baby and skid baby, or skid baby from skid mothers. Then diversity looks like the difference based on their baby's characters. Because wild type baby, this is not baby, like 10 weeks, sorry, this is 2 weeks, so this is baby. So there are alpha diversities in the large intestine in the wild type mice. So they have very low alpha diversity, regardless of the difference of the mothers. Then skid. So they have a little bit high alpha diversity, regardless of the different mothers, with or without antibodies in alpha diversity studies. Then we also have differences in beta diversities, because so wild type and skid, So baby's genotype is very important for use this platform like LIFSI, a linear discriminant analysis effect size. So this is also well-known to identify what kind of bacteria increase or decrease in your comparisons. Then if you do LIFSI, then you can get this kind of data. So please see just middle part. So once again, we have 5 comparisons. Then just this comparison, skid, y type, skid, then we don't see any difference, this NS. So we didn't do any Lexi studies. Then we did Lexi to identify what kind of bacteria increase or decrease. Then Lexi provides you the list of increase or decrease bacteria. Particularly, for example, in this comparison, wild type, and skid. So if you use the data obtained from normal mice, skid mice abnormal model, then let's give you this. So we have maybe 11 genus listed here. Then 8 bacteria are dominant bacteria present in the normal mice large intestine at 2 weeks. Then 3 bacteria are dominant bacteria in skid. Then, for example, if you use like this comparison, wild type, and wild type skip, then Lexi could not find any bacteria increase in normal group. But instead, Lexi find, then tell you 2 different bacteria predominantly observed in wild type skid. That means skid babies nursed by normal milk. So it's okay if you complicate our experimental platform, it's fine, but try to understand. You can further analyze from alpha beta diversity from CHIME. Then if you can find difference in alpha or beta diversities found in the CHIME study, then you can go further. Like using LIFSI, then you can get this kind of predominant bacteria in each group, in each comparison. Then maybe then another thing is that the LIFSI also give you the scores like LDA. So in our experiences, then score is very important because if you have very few scores like 2 or something, a plus minus doesn't mean. So around 2, then the impact is not so strong, so not so high. But if you have like more than 4, then impact is so high. Then you can more precisely understand what the mean of impact I'm saying. So for example, I found that lactobacillus, which is well-known bacteria you are interested in your food science, I found that in the large intestine in the normal mice, lactobacillus is highly present in the normal situation. However, in the skid, immunodeficient babies, they do not have lactobacillus a lot. So this data show you. But you maybe are interested in understanding how much lactobacillus present in normal mice or skid mice. Then you can go back to the original data. Then you can create like this data. So I can tell you how we understand. So we got 11 bacteria from Lexi. Then we just make simple Excel profiling to showing how much percent of lactobacillus are present in normal mice or skid mice. Because let's say tell you that lactobacillus is very predominant in normal mice rather than skid mice. So it should be higher in the normal mice rather than skid mice. But let's say it's true, actually, because if we just get the data right from here, then collect the lactobacillus data, then make this data, then you can see the difference. Because in essence, around 10% lactobacillus are present in the large intestine of the normal mice. But if the baby is abnormal, like lacking immune cells, lactobacillus is less than 5%. So beneficial bacteria are not present abundantly in the lactobacillus, in the abnormal mice. But see, the reason why I said that LDA score is very important. For example, we have several bacteria listed here, but actually this should be important. But the percentage of, for example, Dorea, which is one of genus, is very few. Actually, there are significant difference if we just do the t-test. But the impact is very few compared with the bacteria which has high LDA score. So this is also important data that you have to understand. So say that, I would like to say that you do comparison between like before fermentation, after fermentation, then you can do, for example, Lexi studies, then you can find out like increased bacteria after fermentation, decreased bacteria, high abundant bacteria before fermentation from the Lexi, for example. But try to make raw data, except graph one more time to understand how much bacteria are present in your sample. Then if your target bacteria is like this, like 0.0% something, maybe you'll be disappointed a little. But your target bacteria is abundant, maybe it's very easy to use it, like probiotics or food fermentation, etc. It can be easily adapted for food science, food generation. So this is one of the technique. Then, okay, I'll continue a little bit more. So I did the other comparison. Then this is small intestinal microbiota after weaning. Then 10 weeks, so they develop, then they don't need the mother. The mother is gone. But what we found is that 10 weeks of age, then alpha diversity looks like skid is not good, regardless of the milk antibodies, always bad. But wild type, always good, regardless of the milk antibodies. So before weaning, if they drink milk antibodies or they don't drink milk antibodies, doesn't matter. So wild type mice can develop their own microbiome in the small intestine after 10 weeks. So it's a little bit disappointing for me because I want to emphasize that milk antibodies is very important. They should take it, baby should take it. But if say that milk antibody doesn't, it's not so important. But we can do similar type analysis. Let's see and details that you can show. But finally, I found difference that I really want to get it. So this is large intestine. At 10 weeks. Completely developed, they are mature, they don't need mothers. But if we look at alpha diversities, it looks like totally different between this and that. Because small intestine at 10 weeks, it depends on the baby's genotype. But 10 weeks at the large intestine where lots of bacteria develop the communities, people say microorganisms, microfloras. The alpha diversities in wild type were skid mice, babies, nursed by wild type mothers, which can produce milk antibodies, always high. But wild type baby and skid babies nursed by skid mothers, she cannot produce antibodies, always low. So meaning that even if baby's genotype is healthy, normal, they can produce their immune system by themselves. But after weaning, they mature, they grow. But if they have experiences not getting their mother's derived milk antibodies, they're gonna have problem in the microbiome at the large intestine. So large intestinal microbiome is very, very important. Most bacteria are present in the large intestine, not small intestine. Then I'm saying that if you want to have very important microbiome, then the baby should have maternal antibodies from milk. Otherwise, alpha diversity goes down, even after weaning. Then you can also do registry studies. Then also you can do the t-test. Then maybe you remember that most important comparison for us is this. This is normal mice, skid wild type normal mice, meaning that we analyze baby. Then babies are nursed by normal mothers who are nursed by skid mothers. Then what we found is that in the case of normal, completely fine model, then some dominant bacteria can be identified that should be colonized in the large intestine of healthy offspring. Then particularly this bacteria, Muscicillium, genus Muscicillium, is very abundant in the healthy pups, healthy offspring, when they are growing by getting milk antibodies. They will lose this if they don't get milk before weaning during lactation. So this is the conclusion. I'm sure that this is not a part of the experiment, but this is a good case for me. So I want to say that. So we analyze PAP always wild type. Then of course wild types grow very well, getting normal milk from wild type mothers. Then their microbiome, not at both small intestine and large intestine, are completely fine. Okay, they are very good. Then we have this skid. Then they have problem, particularly 10 weeks. Then they have problem not only small intestine but also large intestine, because this skid unique type of microorganisms are completely different to the normal status. But what we found is that this is very important. Normal mice, normal baby nursed by immunodeficient mothers lacking antibodies. The baby is normal, they can make their immune system by themselves. However, if they take milk lacking antibodies, then they still have abnormal microbiome biotas and large intestine. Then these abnormalities will be continuous, even long time. So it should be related to the healthy. So what I want to say is that if you want to have very good microbiome in both small and large intestine, particularly in large intestine, you should get good milk, which contain antibodies. So this is the end of my talk, but I would like to show you one more slide. Then actually this slide recent to this morning, bioinformatician in my lab send me, so I cannot explain detail. But using the data, then we can perform this kind of networking analysis. So in this case, what we do is like we use large intestinal sample and small intestinal sample all together in normal mice. We plot everything. Then if some bacteria and the other bacteria both increase and which is highly correlated, then you can see a very big line. Then if a red line, then if you some bacteria increase and the other bacteria decrease opposite negative correlation, then you see green line. The big line is very strong correlation. The small line, thin line is very like a very weak correlation. Patient, like if you don't understand, then let me know. Then I can understand. I can answer. Then you can start discussion.
Prof. Nachi,
Hmm?
very interesting sharing, I think. I hope we can do this experiment in Indonesia like that.
Hmm?
Maybe lesser complexity.
Yeah,
Which,
yeah, yeah.
yeah.
So try
Yeah.
to see like Google time to or Lexi. This is maybe first good opportunities for you. Then this many people in the world use this. Then once again, Chime provides you alpha diversity, beta diversity, and also you can just send the raw data, sequencing data, then you can get like bacterial information from the Chime. So they have data.
Yeah, maybe I can uh, imagine that the important step in our answer, or we can solve the thesis. But in our research, maybe still a little exploration to such as in existing condition, like maybe in my research, we take the fresh vegetable like lettuce, cucumber, and explore the existing microbiome in there, not to treat by something treatment like that. Yeah, just maybe as simple as yeah.
You need to compare.
Yeah, compared to in my compost from the feces of the like chicken feces as the nutrition Yeah.
Oil, like waters or food, then you see huge difference, I'm sure. Then if you use same material like food, then like if you use like food for corrected this part and the other part, then the impact or difference is very small. Yeah. Then what kind of aim that you want? Then, like in our experiment, I introduced you in this lecture that I want to show that I want to like say milk antibodies is very important after weaning. So this kind of like research concept is very important. Then once you have this concept, then you can design the experiment. So otherwise, yeah, you will not get like clear-cut data for comparisons data.
Okay, maybe I want also ask the technical something. Yeah, in your data, I see the film and can you show again the slide about the result? Result about the microbiome that compare the phylum, this one phylum
Okay, so we
family,
have
yeah, and genus. Little, yeah, we can get the information there,
yes,
and
yeah, it's very
maybe.
usual, right? Always like 5 or 6 because this is a very good, huge analysis.
Okay, and if we analyze the genus, there are some not assigned the genus, maybe more than most of it not assigned, and others. What is the mean not assigned? There
Yeah,
is no
exactly.
genus identified. That mean?
Yeah, exactly. So some bacteria stop to the family level, not genus, because they don't have genus name. In that case, not assigned. So in the bacterial classification, then people try to give the name, like from phylum, like family and genus, then species. But then the microbiologists distinguish, have to classify. Then in some cases, then they don't classify into genus, just family level. There are so many bacteria. That's why we have to think about what categorized bacteria will be analyzed, like phylum level or family level. If you think about 3, then individual differences and actually significant difference, I don't think you can understand from 3 data. So I'm sure that if you have 10 sample from each group, that would be good data. Then sometimes, so in this case, we are using like 20 or 30, and you can get the result.
Oh, okay.
Yeah, so individual difference is also big factors.
Oh, okay. Yeah.
Yeah,
So we have to have enough sample and data, yeah, for this analysis. Okay, and
exactly. Yeah.
yeah, and how about the from your data standard deviation
Mhm.
in the table? how many plus minus of standard deviation that
Then actually, if we don't have huge differences, then we can get significant differences. Then alpha diversity is three different type of algorithm will be used, have been used in use. Then this is not nothing. That's why people use OR to think about how much accuracy from alpha diversity data was obtained.
Bu Siti, can I ask the question, some questions
yeah,
regarding the
okay. Yeah.
Okay. Okay, thank you very much, Prof. Nochi, for very comprehensive explanations about this. And it actually I have some questions. First
Okay.
is very basic. Yeah, first is very basic regarding the definitions of like Shannon index, observed feature, and fate PD here.
Okay.
Related to the alpha diversity, I would like to know what is the definition of each parameter.
Okay. So I can calculation method somebody make it. It depends. Yeah, but three are well known.
Okay, and then so actually the beta diversity here is actually is similar to this is like a principal component analysis, yeah, if I'm not mistaken, from the figure of the beta diversity.
Yeah,
So we
it's
can see,
PCA analysis.
yeah, PCA analysis based
yeah,
on
right.
the data only.
This is also PCA. So this is
Yes.
beta diversity. Yeah, that's true.
Okay.
So I just break down this part. I took this, then do this. Just take this, then do this. So if you break down the result, like taking this, show this, or taking this, then show this, then the image will be changed, of course.
Yes.
Yeah.
And then, for example, you said that for giving a robust model to like, conclude the data.
Differences, then maybe you will not perform any more researches. So it's not good. So I think like, yeah, always try to get 8 to 10. Yeah, it's good numbers from each
Yeah,
group.
it's a 10. For example, here we can see there are 4 group of treatment. Each treatment give us, if I'm not mistaken, give us 40 samples, right? For
Yeah.
healing.
And
40 samples.
also from small intestine and large intestine. So
Yeah,
it's
okay.
yeah.
And then if in case the treatment, how we manage this, the number.
The difficult question.
Yeah.
Yeah, but I just can say that I try to get more.
Oh, okay.
Yeah,
So we have to
yeah,
adjust, yeah,
yeah.
as many as we
But
can.
yeah, one thing I can suggest you, this is maybe not answer for your exact question, but I just showed the microbiome data. But I think you will also do your experiment in your field, like food science field, like investigating how much like proteins are contained or something like that, or how much taste or something like that. So we can add these data too in the microbiome result. Then also you can reset the comparisons. So if you just do microbiome studies, then only differences like control or positive treatment. But if you have the other data, that will be more powerful. It's like bioscience studies. So not only microbiological experiment, but also many food science data should be collected. Combine it. That's next option.
Okay, I have the next question. Is it possible, Prof. Nachi, regarding the data analysis? For NGS data analysis, can we get the informations to specifically to the species of the microbes? Since I have read from the publications, for example, because gut microbiome is related to different kind of disease, and genus is not enough actually for example, diagnosing disease or to correlate between certain disease with the gut microbiome profile. Could you give us, can we get the species data analysis, the species data in this data analysis for NGS, or
Yeah.
only until genus?
Yeah, actually, yes, it's a good question. Maybe I told you the first previous NGS course lecture, then I said that if you want to identify species, then you should definitely perform a shotgun and whole genome analysis. Yes.
Yeah.
Otherwise, you cannot get species name. If you do this, you can get it. Then you can use your NGS to perform this shotgun sequencing for genome analysis. Yeah. Just difference is the size of the data. So if you do this, then you can't combine lots of sample because this data will be used so many, space in the sequencing system. So yeah, you cannot analyze on the same time together a lot.
Yeah. Okay, I see. Okay, thank you very much.
Thank you.
Maybe question from others audience. Here is the Bu Seflin. Maybe Bu Seflin have already have data.
Prof. Noci, I want to ask about the what the standard for you use for the microbes, RDP, Silva, or green beans. I mean, I join your seminar for second time, three maybe, because first is in in auditorium, and then in seminar yesterday, and this I never knew that what is a standard that you use. You use RDP or Silva or green gains or what? In the so you can choose that you have a species this, species that, species that this something like that.
What's the mean standard?
Yeah.
Database
Especially if we use SIME, we
Hmm?
use standard is maybe you use green beans, silva,
Okay,
RGB.
Got it. Okay. So basically, we are using most of the things. Then try to compare. Then, yeah, so this is maybe the options. Then, of course, the database have a little bit, it's not exactly the same. So if you have concern, then you should use everything. Then try to get very accurate data. So this is very common approaches to perform NGS experiment.
What can you repeat?
Yeah, so I know that, yeah, we agree that like there are so many database, like standards you said.
Yeah,
Then
database,
yeah,
standard database that you use.
yes, then we agree to use several different of database because one database has like this part of positive, but the other database, this is also a good thing. So we have to appreciate those everything. Then in our experiences, we basically use most of the database to get the data. Then if data not be changed, the data is true.
So you don't use silva, you don't use RDP, green beans. You
We use those.
But so you use all of that standard
Yeah, yes,
database.
exactly. Yeah.
Oh,
Then compares.
okay.
Yeah. So it's kind of bioinformatics approach. Then we want to have 100% accurate data. So if there are some tools, then we try to see it. So this option bioinformatician in my lab always doing.
Okay.
Yeah. Many people are trying to make like platform, database, software, etc. The most of them are free. Then bioinformatician in the several laboratories, including my lab, is always checking what type of new is developed and then uploaded in the database or in the web system. Then we're just checking. Then there are some kind of communication too, discussing about what's new. Then bioinformatician always checking about what they are discussions. Then sometimes they found that this is very new platform. They always take it, then try it. But this is mostly free, so everybody can use in the one.
Thank you.
So we can conclude that when we use KIME as a platform for data analysis, we can adjust the database to be used as a reference during the analysis of genus, right? Oh, okay, so we can adjust which database that we
Yeah.
would like to choose.
Yeah.
Yeah. It is almost similar to the previous one, ya, Bu Evelyn, as mentioned by Pak Nanda. So we can
Ya,
adjust
biasanya mereka
database.
kalau pakai bakteri itu biasanya bisa pakai silva, pakai RDP, tergantung dari standar database-nya yang terbaru. The newest one is which kayak I think Prof. Nochi maybe he used many standard database. I don't know.
Yeah. The point here is that if we use different database, then if data will be changed, then maybe we'll have problem in the future. Then somebody noticed that our data is not correct. Then if we use different way, then data will be changed. That's the problem. It's not paper. So yeah, we have to check. It's our responsibilities always.
Yeah, okay. Thank you, Mr. Nochi.
But if we have huge difference, then database doesn't matter, I think. Small difference always make problem.
Maybe I have one more question, Prof. Nochi, regarding the use of NGS platform for transcriptomic analysis. Do you have any experience with it? And do you find any critical point that we would like to do the transcriptomic analysis for in using in NGS analysis? Is there any critical point that we have to like pay attention to?
Okay. Yeah, actually, I'm thinking there are several critical points in the NGS, but like DNA preparation PCR, it's very well established. I don't think this cause many problems. But most problem is sample correction or how we extract DNA. That's why I had a call. I had a chance to tell you about importance in the first lectures. If we choose miss a wrong sample correction material or procedures, data will change. But if we can get DNA, then we can use PCR. Then 16S ribosomal RNA PCR is very easy to get banked. So yeah, you can run sequence those. Then you can do like omics time-to analysis. But think about that. If you are having wrong sample, not exactly corrected in your from your sample, that's more problem. It should be standard.
Thank you.
Prof. Nochi, I interested the last slide.
That's one.
Last slide.
This one.
Yeah, I think it's very
This
interesting.
morning data.
Yeah,
I can't
yeah,
explain.
yeah.
I need to ask my bioinformaticians.
Yeah, what kind of the software you use to
I'm sorry,
get
this is the information I didn't get it. Yeah, but my bioinformatician is always searching. Then this is also one of free software, free online platform. Yeah.
Mr. Noji,
sorry?
do you use stethoscope for this
Salah,
visualization?
tolong panggilin Elvin dong.
Prof. Noji,
Hmm?
you use stethoscope for this visualization, data visualization?
I don't remember the name. I usually don't care.
I think it's cytoscape. I think it's
Okay.
we can make what a connection in this is about connection. I don't know. There is no title or no
Okay. Thanks.
I don't know. I think it's you use cytoscape to connect between gene and gene, something like that.
Yeah, I think principle is same. Like bacterial studies, gene studies, yeah, principle same, I think. Yeah, you're right.
I don't know what the title is. I
So
just know
sorry.
that you use cytoscape, but I don't know what the title is. What is connection about?
Is any paper?
Once again, try to get the data from your field and combine the data with microbiome. That is very important.
The interaction about the gene. Yeah, that's right, sir. Yeah.
Like in your case, like we are always collecting, like for example, plasma, then checking cytokine levels, then we add everything. Because some cases, if we make two groups, like positive group, negative group, but the data is not positive, like expected. But some mice, they produce very high cytokine even in the positive group, even in the negative group. But we want to excluded the data because we cost. That's why we remake the group one more time, like cytokine positive group, cytokine negative group. Then you can get more important information. But yeah, in some cases. So don't discard the data.
Is there any your paper that publish the figure like that so we can read
Oh yeah, so
yeah.
maybe So yesterday I talked to you in the conference. I'm not sure who attended. So let's show. So this is one of the papers. Then yeah, we are doing fecal microbial transplantation. Then we are trying to understand how fecal microbial transplantation works well. Then we did the microbiome studies by tracing the change of microbiota. So this is one paper recently published. But this journal, microbiome, is very good journal, so you can take a look.
Okay, thank you. I will try to read.
Yeah.
Is this open access paper?
Yeah, yeah,
Okay.
It is.
yeah, you can get
James.
it. Yeah, if you have a problem, let me know. Yeah, I can email, I can send this. Anytime we have a connection, so anytime email me, then I'm very happy to help you. Yeah. So that,
Okay.
it's okay, for example, then we are very happy because this kind of NGS, where when we do NGS studies, it doesn't matter. We can share the data, then we can analyze. But so if it's okay, then we are very happy to support like analyzing. We have bioinformatician, then sending back to you, but we are so yeah, this is very large scale collaboration.
Okay, thank you, Prof.
Yeah, yeah, yeah.
I am we are very happy to
Yeah,
hear
yeah.
that.
I'll have more chance to visit Indonesia. I'm trying to make my intestinal immunity much stronger.
Yes, of course.
But I got something, so my immune system makes very strong now. So yeah,
And how was your condition right now?
yeah, much better. But this morning was very bad. I'm still so sorry. So I couldn't get up. So
It's okay.
suddenly happened.
Yeah, I heard you find out the lactobie powder in like the probiotic powder.
Hmm?
Maybe in Indonesia we call it lactobie for
Lactobacillus. Yeah, lactobacillus.
stomach.
Hmm.
You already get in the store or no?
Yeah, we have several yeah. So we are very interested in utilizing those for our animal studies. So yeah, it's one of our target. Yeah, it's true.
Okay, from Bu Ines.
I have question.
Yeah, sure. Hello, hello.
Yeah, hello, Nachi Sensei.
I'm so sorry I couldn't see you.
Yeah, me too. I'm so sorry to hear that you getting
Something.
disturbance. I like to know. Sorry, not on gene. I like to know your opinion about if somebody drink Yakult. Do you know Yakult drink?
Yakult?
Yes,
From
Yakult.
Japan. From Japan.
Yes, but we can have here.
Yeah, I know.
But if we drink every day 2 bottles every day regularly, is it good or not? Yeah. Sorry. Thank you so much.
It's good question. The many probiotics or Yakult-like companies said that we should drink it continuously every day. This is company is good if the people buy it every day. But people say that Yakult is good. I don't want to say that Yakult is not good. Yakult is good. But some people can use Yakult for developing their good microbiome. But maybe some people are not taking Yakult, generating their microbiome. So it depends, right? Communicate collaboration between Yakult and your microbiome. So continuously taking the microbiome or Yakult like drink is good. If you take it, then it facilitates developing your microbiome. So you have to test by yourself by checking your intestinal condition.
Yes,
If you,
in my case, I cannot drink Yakult every day because
but
getting
don't
diarrhea.
drink. If you don't drink it every day, it's just juice. That's probiotics. Yeah,
Fruit juice, you mean?
it's like orange juice. So,
Mm.
Yes,
yeah,
Mm.
thank you. I got.
yeah. So if you take continuously, then microbiome is stimulated by Yakult or Lactobacillus, then making it better.
Okay, thank you very much.
In here also there are lecturer from the biology, Bu Andi Ernawati. Is there still any in Zoom, Bu Erna? Oh,
Are you asking me? No, no. Oh, okay.
There are no question. It's now.
okay.
Yes.
Thank you, Bu Erna. Maybe Bu Ines?
Thank you very much. I sincerely apologize my condition, but much better now.
Yeah, okay, thank you. Prof. Nochi, we have another agenda about the manuscript coaching from the student, and I have received 2 manuscript that are ready to submit, and only for the still in proposal, maybe for design experiment. So it's okay if we send first the document
Sure.
before Monday. Monday is the agenda for coaching. We will send first to your email. It's okay, or
Yeah. So
only Monday in
yeah,
Monday.
is it Monday?
Yeah, Monday is scheduled for coaching.
Okay. Sure.
But
Yeah, I
we
can take.
will send first. It's okay. But you prefer to
Yeah,
send for the document.
yeah, just send it to me.
Okay.
Yeah. So tomorrow's I will go to IPB, right?
Yes. Class with Bu Hanifah. Yeah.
Yeah, it's same place, right?
With Burati
Bu
actually.
Rati. Yeah.
Yeah. But tomorrow, not in Darmaga. The campus is in next to you. Like
Yeah.
Baranang Siang campus is next to Santika Hotel.
Okay. So I don't have any more chance to visit IPB, your campus. Monday is there. Okay.
Sorry, I repeat again. Saturday in Baranang
Saturday.
Siang campus for lecture. From S2 program, food safety master program. And Monday in Dermaga campus for manuscript coaching.
Okay.
Bu Hanifah, tomorrow Saturday in what time?
Tomorrow at 9 a.m. But I hope I can be there with you in Baranang Siang. But actually, I have a lecture in the afternoon. Actually, I'm not yet preparing final in a final form. So I hope I can join tomorrow morning with
Okay.
you.
So what time should I be tomorrow morning? Somebody take me up to go to the place.
Yes, I think should be somebody to
Yeah.
Yeah.
I'm happy.
Yeah.
Then I just want to know
Maybe one stop we'll assign to pick you up. Maybe Mbak Dila. Yeah.
what time.
What time, Bu Hanifah?
The lecture is start from 9 a.m. So Maybe
9 or 9:40. You can make sure, Bu Hanifah. 9 or 9:40.
Pak Pungki want to change the time, so can start from 9 a.m.
Okay.
not 9:40.
Start from at 9, so the staff will will pick you at maybe
Maybe at
8:45.
8
berapa?
because
At 45, at
8:45.
yeah,
Okay, so
15 minutes enough
okay.
walking from Santika to
Yeah.
the
So I will be in the front entrance of the hotel at 8:45.
in lobby ya, in lobby.
Okay, okay.
Yeah, still inside. Ya, mungkin Mbak
First floor, lobby floor.
lobby.
Okay.
45. Yes.
Ya, Bu Hanifah mungkin nanti ke Dila ya, Bu Hanifah yang ke
Iya,
Dila.
tapi
Ya.
ada kemungkinan Bu Falel, ada kemungkinan saya, tapi nanti saya lihat dulu kondisinya. Saya soalnya belum preparasi dengan baik yang besok. Besok kan jam 1 sampai jam 5 kelas
Ya,
saya.
ya.
Cukup
Kalau
banyak.
Falel kayaknya nggak ada Kali, nggak ada di
Bu
lokasi.
Falel yang ditugasin oleh Bu Rati sebenarnya.
Oh, oke, sorry, iya nggak ngikutin.
Bu
Nah,
Falel ini kalau saya bisa datang pagi,
ya,
maka Bu Falel enggak
oke.
begitu. Cuma
Oke.
saya nggak bisa mastiin, nanti lihat kondisinya
Oke,
besok.
ya, yang penting kan Falel berarti yang sudah di ini ya. Kalau nggak nanti saya juga minta Dila kalau misalnya tidak Falel, gitu ya Bu Hanifah.
Kuliahnya masih dipersiapkan ini.
Ya, oke, nggak apa-apa. Yang penting ada satu aja yang di-assign gitu, Bu.
Iya, betul.
Pastikan ya. Saya juga akan kuliah sih sebenarnya tapi jam 2.
Oh iya, saya mahasiswanya sama KPM. Ya.
Ya. Baik, Bu Ines silakan dipimpin lagi. Bu Ines yang mimpin.
So 8:45.
Ya, 8:45.
Okay.
Yes, someone, Valerina or Dila will pick you in lobby.
Sure. Thank you very much.
Bu Ines enggak?
Once again, sorry today.
Oh, suaranya ya. Bu Indah suaranya nggak ada ya? Nggak usah pakai ini kali ya?
Can you hear me? Prof?
Ya, oke.
I actually want to ask about if our sample have a lot of not assigned data, is it actually bad or like the extraction is not good?
So all sample data is good, not good.
Mm.
So what's the meaning of sample or data bad?
The question is
So
if we can find a lot of the proportions of
Not
unassigned bacteria.
sign, yeah.
But this
Yeah, I think it depends experiment. Then actually it's just bacterial classification. Then if you have like unfamiliar bacteria a lot, which do not have like genus level, then it means they're non-assigned. But you can see that how many bacteria-derived DNA have been sequenced in NGS. Then if you have like very high number of bacterial DNA, like 60S ribosomal RNA, then you did exactly. Then, but if you are sequencing data, then if you get like non-bacterial sequences, then you did something wrong. Because your sample contains some non-bacterial gene. So purification, extraction DNA after PCR is very important.
Thank you. That's all. Maybe anyone else want to ask? Okay. Sorry.
Maybe one more last questions, maybe from
Sure.
while waiting for other participants to ask the questions. Yeah. If we work with the gene expressions in using NGS platform, can KIME is used for the platform to data analysis, or we can use another kind of platform that is available online and it is freely accessible for the gene expressions?
Any platform can be used. It depends on your research purpose.
So it is KIM platform is not only for microbiome, right?
Yes, that's true.
Okay.
Correlation means, for example, if you want to use correlation studies, then many data, not only from microbiome, but also the others, will be combined. So it's not just for microbiome.
Yeah.
Okay. Prof. Nochi, some of us use or have already learned about the Galaxy software. So what is the differences between Galaxy with Key?
Mm. Is that okay?
Oh, I waiting you answer about the difference
sorry.
sorry,
I'm sorry.
between Galaxy open source and the key.
Yes.
Is different as any different function or different model or different result.
Well, but it's very well known, so yeah, I try to use it first. Yeah, it's only my suggestions.
Okay.
Sorry, I missed it. Yeah.
Yeah. Okay. Thank you, Romi. Sudah, Bu Ines.
Can conclude today training, Prof. Nochi. So,
Yes.
I think we can conclude our meeting here. We already a lot of things to discuss. If there is anyone else who want to ask, I think this is the end of the meeting today. Thank you very much for.
Thank you very much. I'm so sorry. Yeah.
Maybe you can give photo session, Bu Ines.
Sure.
Yeah.
Wait. Okay. So I will take the picture.
Good.
1, 2, 3. And there is another slide. 1, 2, 3. Okay. Thank you very much.
Thank you very much.
Okay, thank you very much, Prof. Nochi.
Thank you.
Yeah,
See you tomorrow. Bye.
Yeah, bye.
Bye. Thank you, Prof. Nochi.